I'm having trouble sharing today.
Sounds like my 6-year-old daughter.
No, that's wrong view.
You can just
All right, I think I got it now. There we go. So hello, everyone. Welcome to the meeting. As usual, this is the agenda. We'll kind of go through an executive summary, and then I'm going to turn it over to Emily from CAMH, and she'll be discussing our collaboration on using ALK2 inhibitors as PET imaging agents. Then we'll discuss some of the activities that have been going on since we've nominated M4K 2009. Is there an issue? Oh, I thought somebody said something. And then finish off with some next steps of planning with M4K 2009 moving forward. So since the declaration of M4K 2009 as a development candidate, we've been working on getting some additional ADME and DMPK studies, so those are up and running. And we've been trying to work through getting some CMC contracts in place. So for the salt screen, we're in process of getting the master services agreement in place. And we do have a live RFP for scale-up and analytical method development. So we're expecting to get proposals in from that probably in the next 3 weeks. And in the middle of December, we'll be evaluating those proposals and choosing a vendor for scale-up. At the beginning of November, we met with this advisory committee as part of the Tessa Jowell Accelerator program. And I think that meeting was very useful, and I'm glad that we went. We received, let's say, positive feedback on the program, and afterwards some offers from the people there to help out and with the program. And I think they saw value in what we're doing, and really I genuinely think they want to help us succeed in this program. And we'll get a full report from them in early January on the review of the program. We did discuss the hERG value of M4K 2009 at that meeting and over the past month. And the reason for that, we put the program in front of a potential investor, and they declined the program. One of the reasons was they viewed our hERG value as a tox liability. So we've done a lot of kind of due diligence internally and asked, expertise outside of our program for additional feedback. And I think, the feedback we got is this is something we need to be aware of. We certainly are aware of it, but to continue to push forward with the program and eventually discuss this with regulators is, based on the safety window that we've calculated for 2009. And then we also had some discussions with the Connect Consortium around what a potential clinical trial is going to look like. So we did discuss a few potential scenarios, but ultimately, we came to the conclusion that we need to really begin the process of setting up a meeting with the regulators to discuss our clinical plans. So we're going to start working towards that, but we'll also get some feedback from the Tessa Jowell Foundation on what a clinical trial might look like. So I'm going to stop sharing and turn it over to Emily to discuss the PET imaging project.
Thank you. Let's try. Does that work for everyone? Okay. Yeah, so if we haven't met, I think it's been a while since we've kind of given an update to the greater project team on what we've been up to at CAMH. But I'm Emily. I'm a staff scientist, radiochemist at CAMH, and assistant professor in the team there. A lot of the work that I'll be showing today has been led and performed by one of our PhD students who just defended last week, Melissa Chassé. So get into it. I guess, again, I don't know people's backgrounds, but most people are usually familiar with PET as kind of a clinical imaging technique for diagnostic imaging. In this case, on the left here, I'm showing a diffuse glioma where you can use either FDG, so like sugar metabolism, or amino acid transporter PET imaging for glioma imaging. But then on the right side of the screen is more where we work within PET in research and in clinical trials, where you can use PET to stratify patients based on whether or not they express a biomarker of interest that will respond to your targeted chemotherapy, or we can use PET to measure efficacy of drugs to help with dose projections and target engagement, especially for CNS PET, where there's no peripheral readouts of the measure of your target engagement. So positron emission tomography, PET essentially works where we make a radioactive isotope in a cyclotron, or sometimes a generator, and then we attach this radioactive isotope into a drug molecule. We inject the drug molecule, it circulates in the body, accumulates in your target of interest, and then within the scanner cameras can pick up the emission of the radioactivity, and then we reconstruct that into our images. For neuroimaging, we usually work with carbon-11 or fluorine-18, which are fairly short half-lives of around 20 minutes or around 2 hours. And we are working to make these radioactively labeled drugs within around 3 half-lives of the isotope production. So the chemistry and tracer production is done on demand for every kind of single PET imaging study. So for this project specifically, and our involvement in this and the pre-CATA grant that we have with the OICR was split into mostly 3 different aims. Aim 1 was to evaluate whether the drug candidates that the OICR and others were making have adequate and even distribution into the brain and into the pons, so we can PET isotopically label the exact drug molecule with a PET isotope like carbon-11 and give it into animals and see how much of it and whether it's actually getting into the brain or not, and also whether it's then getting into the pons. Additionally, aim 2 is where we've been working largely lately, is in trying to develop a radio tracer that's ideal to measure the target engagement of a potential drug. And so we can measure a receptor occupancy of the drug candidate. And then aim 3 is kind of a more explorative aim where we're trying to see if ALK2 could potentially be an imaging biomarker in order to do that like patient stratification that I mentioned, or if we could turn our drug molecule into a radiotherapeutic, which is kind of an advancing field where we would isotopically label the drug molecule with a different kind of radioisotope that emits a therapeutic kind of radiation. So the initial proposal looked a little bit like this. When Ian Watson was with the OICR, he approached me when I was a postdoc back at CAMH, where he brought to us some of the lead compounds from the OICR that had promising kind of parameters for the drugs, including 2009, obviously, as well as some other ones that we thought maybe would have a better brain to plasma ratio, or better brain permeability, but maybe different affinities for ALK2. And so carbon-11 labeling of these, this is what it looks like for pretty much all the structures I'll show you today. We have to do this behind lead shielding, and it's all automated and remote controlled on a kind of microfluidic almost system. We take carbon-11, carbon dioxide that's generated in the cyclotron, and then we convert it into methyl iodide or methyl triflate, which then we can use to methylate precursor molecules. This then goes through semi-preparative HPLC to purify them. We reformulate them into an injectable dose and then put them into our rodents at CAMH. So mostly we have been labeling either in the bottom ring at that N-alkyl site or in the top ring on one of the methoxy groups of these kinds of structures that we've been working with. And so this was the first, again, 4 that we talked about. These are different, I guess, images and versions of the data that we acquire in PET imaging. So on the right, you'll see what we call time activity curves, which is basically on the y-axis a standard uptake value, which is a measure basically of the radioactivity that's been kind of normalized to the injected dose and the animal's weight and the radioactive decay. And then that's over the time of the scan. And then on the bottom, you see kind of our heat maps of the radioactivity distributed in the animal by standard uptake value again, where we have the brain of the rats here circled with the white dashed line. So we were, we like the radioactives, the activity, so the structures all get into the brain, some more than others as a tracer structure here. But importantly, whether they got into the brain or not, really, we saw that they were evenly distributed into the pons as the rest of the brain, which was important and kind of what we were trying to prove here. Surprisingly, 2009 had the lowest uptake out of the ones that we were looking at, and 2127 had a higher uptake. And usually with PET tracers like this, we're looking kind of for SUVs greater than 1 as that initial peak uptake. So we decided after this to kind of move forward with evaluating M4K 2127 a bit more as a PET tracer. So as I mentioned, for determining like target engagement and drug occupancy, we need a good PET tracer, which is like has different properties often than a drug molecule. And so there's optimization of the tracer structure and characterization of the tracer itself in order to be able to do this. But the target engagement study would look like this, where we do a baseline scan and we get our PET uptake, and then we would administer a drug dose at like a therapeutic dose. And then we would redo the PET scan and see how much that PET signal has decreased at that dose. And this over multiple different doses can help derive kind of an in vivo directed measure of like an EC50 or ED50. But as we see, like the PET scan only really sees the radioactivity, so we really need to characterize really well what this tracer is actually doing. I won't go into like full details of this slide, but there's different things to consider for tracers in addition to kind of drug properties that you would look for to make sure that your tracer is getting into the brain. But because we're dealing with short half-lives, and so kind of a short scan time after the tracer has been made and injected, the tracer has to be passively brought into the brain. And so it has to have decent lipophilicity, but also not be a substrate of efflux transporters. And we kind of usually want different kinetics, so a bit of a faster clearance than is designed for most drugs. Other things that will come up a bit in the next few slides are we're looking for something that has high target selectivity. So we need low non-specific binding. So whether or not, if the tracer is sticking to kind of other things in the brain, aside from our target of interest, we still see the radioactive tracer there. We need to make sure that our signal is coming from on target to ALK2. And we also need to make sure that that radioactive signal is not composed of metabolites that still contain the radioactive tag. So we, going forward, was characterizing M4K2127 a bit more. We did first what we call a blocking study, which is basically pre-administering the unlabeled version of our drug before the PET scan and seeing how much that decreases PET signal. And our first time that we did this at a couple different doses, we didn't see any decrease in our PET signal. And so it was confusing kind of initially. And so we wanted to figure out why that was happening. So we looked at radiometabolites as well. And what we found was that only 25% of the signal in the brain was actually our intact tracer, and the rest of it was carbon-11 methanol, which was getting into the brain and obviously not able to be blocked by pre-administering our unlabeled compound. So we did a few other studies in addition to this and tried a couple different other labeling metrics on that and on alkyl site, but we ultimately decided that labeling on the N-alkyl position was not going to be viable for these structures due to radiometabolites that were arising. So again, we tried a few different things, but the next kind of step in our process that we got to was we ended up moving our labeling site up to those methoxy groups in the top ring, and we also ended up kind of capping that amine in the bottom ring with an isopropyl group to improve our brain uptake, as we saw that the free base versions were not very brain permeable as a PET tracer. So these had better brain permeability and no metabolic liability, but we still kind of have fairly low brain uptake as far as PET tracers go, with that peak SUV being less than 1. And the only ones that you see on the right, on the graph on the right there, that had peak SUVs greater than 1, most of that activity was actually those radiometabolites that I mentioned. So conveniently around the same time, OICR was publishing on these cyclized structures that had really high reported brain to plasma ratios that we were quite excited about, and we hoped that labeling these would give us brain PET tracers that had really high permeability. And so we labeled the first couple of them, namely 2308 and 2304, and we saw actually exceptionally low brain uptake of our tracers. But again, importantly, all of this tracer that did get into the brain was evenly distributed within the pons compared to the rest of the brain. We wanted to probe this a bit more. And similarly, at the same kind of time as we were discovering this, I was on one of these calls and I saw some MTCK data that I was concerned about as far as PET tracers go. It's one of our biggest hurdles, I think, to developing good CNS PET tracers, is that they're really prone to efflux transporters because we are dosing them at a tracer level. And so if there's kind of any efflux that they're subjected to, it's very hard to overcome at tracer doses. So these numbers were concerning to me that we were looking at. And so what we did was tested them in our rats. We pre-blocked the rats with alacridar to block PGP. And we saw quite big jumps in our brain uptake across 2308, 2304, so the free base versions, as well as anisopropylated versions that we had made to see if that improved the baseline uptake. And so this is kind of the end of those tracers that we decided as PET tracers, these cyclized compounds weren't going to work for us because they had too much efflux liability, but it was a good lesson for us. And so we've moved now into designing other radio tracers that we think will have less efflux liability, including the ones shown on the structure that have um, less hydrogen bond donors and kind of different pKa's involved in them, as well as different polar surface areas to really optimize that passive permeability into the brain. And so these tracer or these time activity curves at the bottom, we've moved away from doing the alacridar in rats and instead have purchased some triple knockout mice. So they're missing the MDR1A and B and BCRP efflux transporters. We've been using those instead to kind of compare whether or not these are substrates of efflux transporters. So 2209 was still, it was the next tracer that we evaluated, and again, the baseline, that orange line didn't have great brain uptake. And then our newest one that we've been looking at is 2119, where we have that peak SUV that we're looking for that's greater than 1, and a good kind of kinetics of that curve of the tracer washing out. So we're really excited about these new generations of compounds and hoping to reduce that efflux liability and create a good radio tracer. So this is summary kind of where we're at there, where we have no metabolic liability, good brain permeability of 2119, reversible tracer connections. But the next thing to test is whether these tracer signals is specific and selective to ALK2 in vivo. So I'm showing the data here just with 2119 because that's the data that we have so far, but with the caveat that this data is in those triple knockout mice so that our brain signal is high enough to really measure and displace. But we do so far see a dose-dependent blocking if we pre-administer with the unlabeled 2119. And then we also see some blocking using 2009. But we still need to make sure that this signal is selective to 2009, or sorry, to ALK2. And so that'll include doing some urine safety screens and doing some ex vivo competitive radioligand binding studies with other drug molecules for different targets to make sure that it's specific to ALK2 still with 2119 or other tracers. And so, yeah, so this is kind of the overall goal. To go back to this slide that I showed earlier, is using one of our M4K tracers to demonstrate target engagement and to support dose projection for M4K 2009 for an IND. And then aim 3 is an explorative aim. We've been looking at whether or not we can use ALK2 as a diagnostic PET biomarker. And so importantly, like overactivation of ALK2 is not necessarily protein overexpression, and increased gene expression of ALK2 is not necessarily increased protein expression. So we're doing some autorad studies or autoradiography studies with tritiated versions of our tracers to explore ALK2 expression in healthy pons tissue versus DIPG tissue. And so far we haven't had very promising results, but again, there's some caveats to these studies that the tissue is obviously not age matched. The sections of DIPG tissue that we have are very small and have some signs of freeze thaw, so they might not be the best quality for these kinds of studies, but it is the other aim that we are working on within this grant and project. And so in summary, I like to reinforce to medchem teams that good drugs are not necessarily good tracers. So none of this data is against 2009 or any of the other structures that are part of the therapeutic program. Importantly, small structural changes can really affect our radioligand distribution, and we have kind of different hurdles of developing a good tracer than those encountered in medchem. But ultimately, we hope to be able to show target engagement preclinically to support an IND and potentially clinically within a phase 2 program. And also determine if ALK2 does have differential expression, if we can use our radio tracers for diagnostic imaging or to develop into targeted radiotherapeutics. That's, I think, it for today. But if you have any questions, I'm happy to help, or if anyone wants to reach out anytime, I'm happy to discuss more.
Thank you. I have a quick question, actually, two quick questions. One is I want to make sure that we all understand that the dose you are using for this tracer are relatively low for what we compared to what we would normally use for our efficacy and other kind of doses, right? Like PKN for the compound that is going in the clinic. For example, we'll do dose like 10 to 30 milligram per kilogram, but you are going much lower than that, right?
Okay, that's helpful information. We did kind of start at I think some of the dosing that we had PK data on for our blocking studies. For if that's what you mean, we're using like the 1 mg per kg, because that's what we had data on for blocking. But I don't. Obviously, if we were to do a full receptor occupancy curve, we'd go up to the higher doses that you're using as therapeutic doses and around there.
Okay. Yeah. Okay, good. Thank you. The other thing I was going to ask you, like, did you do the radio labeling? And maybe David Smith can also answer that on M4K 2045. That will be the close analog of 209 with the isopropyl group at the bottom. 2045. No, you're muted, Dave.
Yeah.
I don't remember offhand if we had the precursor for that and if we gave it to CAMH. I'd have to go back and check for that one.
Yeah, why I'm asking, because like now you have a two cyclic ring. I mean, you have the aromatic ring and the cyclic ring at the bottom, but this will be the close analog of 209. And then I was just curious because 209, the other ones were good. Like if she go back to slide 11, only thing different would be the isopropyl group there.
So you mean 2119?
2045, I could see from the M4K.
Sure. Yeah. I think that was on our list at one point, and it got a bit deprioritized when we were working on the cyclized structures. But I think it is still one that we can evaluate next. It's worth trying. Most of these structures haven't really shown much brain uptake for us, but we definitely can include that.
Great, thank you.
Yeah, I had a question if nobody else does. Are we sort of breaking new ground with intracellular targets, or have radio tracers been successfully used with intracellular targets versus cell surface targets?
Intracellular targets are common, not like super common, but they are used a lot, especially in CNS imaging. I would say kinase targets are really tricky. There's not a lot of ones that have been actively done for kinase targets, just because especially getting that selectivity and specificity is a lot trickier, obviously, most of the time. So it's not entirely new ground, but it is kind of the thing that it's hard. We don't have a lot of precedence of knowing what makes a good kinase target tracer.
Thank you.
Hi, Emily. My name's Mike Briggs. I'm a DMPK scientist. So I've got a question around, with the slide in front of us here, you've got your SUV, and you've talked about having a nominal target of at least 1 for a good ligand for target engagement measurement. How does that type of information, what's your understanding of how these numbers translate into CNS distribution into the clinic? And if we're looking at a compound at this point with an SUV in whatever assay this is of less than 1, what are the sort of chances of success in terms of CNS distribution in patients, for example? How does it translate from a rodent through to human?
That's entirely fair. I mean, yes, the SUVs are like a type metric that's used so that it is kind of more normalized theoretically across species, and because it's based on the kind of proportion of injected dose and body weight and stuff like that. I guess like the biggest caveat here is that yes, if most of the reason that these SUVs are quite low is due to like PGP efflux, which is more common in the rodents, then there's potential that these tracers would perform better in humans.
Okay.
Which has less PGP. So there is potential that these could be higher SUVs in humans and would be able to do a target engagement study in humans.
Okay, so is that something you've seen in the literature or have got personal experience of how projects have progressed from preclinical into the clinical, and you've seen that improvement in CNS penetration for something which was nominally or putatively an efflux transporter substrate? That's sort of useful information, I think.
Yeah, there's a lot of examples, I guess, kind of both ways, where sometimes tracers that don't, if you can really narrow it down to the reason that it's not working is because of the PGP flux or something, then there's more reason to translate it into humans. But sometimes convincing someone or a site to do that translation for a radio tracer that doesn't look very good is tricky. I mean, I've also seen it go the other way, where something that works really well in rodents doesn't work in humans or higher species because of like higher BCRP flux or something like that in those species versus the rodents. So I have seen it both ways, where something fails. I mean, we had a tracer recently that looked really good preclinically, and then in humans, it's like plasma binding was much higher, and so there was less free tracer to distribute into the brain, and so we've kind of had a poor tracer in humans for that metric as well. So yeah, theoretically, the numbers should translate across species, but there's all those small things that do come into play.
Another thing, I mean, it's just for yourself. I would say at any time when for a project like this or any other project, just ask for any upfront metabolism biotransformation data on any compounds. I don't know if you had access to any of that for these studies, because you talked about some of those N-methylated labeled compounds, and you found that 80% of it was methanol. But we'd sort of discounted some of the compounds earlier in the project that looked interesting from a CNS penetration point of view, but they were heavily cleared by that particular route of metabolism. So it's just the sort of, try and grab or ask sort of teams, what have you got? How, to help understand the most sensible site of labeling in terms of metabolic stability, I think would be useful.
Yeah, of course. It's evolved over time as we've gotten more involved in the team and been able to get more of that data.
It's very interesting.
Just one comment from the chat from Melissa. I guess on slide 19, she just wanted to clarify that this BPND blocking histogram was actually with M4K 2209 and not 2119.
Yeah, sorry. I apologize for that.
Any additional questions for the CAMH team and Emily? Yeah, thanks you so much for putting that together. I think, at least for me and probably a lot of the other chemists, it's been interesting to be part of this and kind of learn the subtle differences that you have to think about in the properties of a molecule of a tracer versus a drug. So I think you did a nice job of kind of going over that on this presentation.
Thank you.
Okay, so in terms of the agenda, we'll move on to some recent activity with 2009. So I mentioned at the top, we went to this Tessa Jowell Foundation meeting in early November. Ahmed and I attended in person, and some other members of the team joined virtually. And just as a reminder, this was, we were enrolled in this program and we're getting essentially free independent review of the program from experts in different areas. So some of the feedback that we got from the group was that they were really impressed with our unique approach and the expertise and the team that we've built for this program. We did discuss, I guess, a number of topics, including the HERG and safety, which I kind of mentioned at the beginning. They really wanted us to focus on looking at additional combination studies, which certainly we're considering, and with the knowledge that this is not going to be a standalone agent. Talked about some regulatory hurdles that we might encounter along the way. Discussed the open science model and the regulatory implications of that. But we also had some input from patient advocacy, and I think this was a really important aspect of that meeting, where they really want us to have that inclusion as we think about progressing this compound into the clinic. It's really having solid feedback from patient groups and patient advocacy. So we'll be getting the full feedback from that review, hopefully by early January. And once I have that, I can share that with everyone. Does anyone have any questions or comments about this meeting that we had? Okay. So then before I turn it over to Liz, I just kind of want to go through this whole discussion process that we've had with HERG over the past month or so. So as I mentioned, we put this in front of a potential investor, and one of the reasons they declined to kind of move forward was this 12 micromolar HERG as a potential tox concern. And I guess in some organizations, they have a cutoff of greater than 30 micromolar. We've always gone through with a cutoff of greater than 10 micromolar. So, we discussed this with a number of different experts in the field, and I think the feedback we got is, okay, yes, this is something that we need to be aware of, but it's not a showstopper. So push on and go in front of the regulators with a calculated safety margin. So when I was looking back through how we kind of derived this 12 micromolar value, it came from two runs that were done at Charles River. So we had a value of 18 micromolar that was done early 2024, and a historical value near the beginning of the program of 8.3. So that's where we calculated that value. But there were two additional runs that I found, one earlier at Charles River, and then a more recent one that we ran earlier this year at Pharmaron. So if you kind of take the geomean of all four of those values, we're in the range of 9 micromolar. So I think it's just a question of how do we frame the actual value that we're going to kind of report moving forward. But in the end, I believe all of this is manageable, and Liz will get into that on the next slide. But we have at least two compounds that are in clinical development for DMG, and we see that both of these have low micromolar HERG values. So obviously they've been able to progress those compounds into the clinic, and I'll turn it over to Liz to discuss our strategy for calculating a safety window for 2009.
Thanks, Peter. So we kind of went through the human dose predictions and how we kind of approached them a couple of meetings ago. I think what we really wanted to do here was look at what we have predicted for human dose and look at the Cmax and how that compares to the HERG value that we've derived from the studies that Peter was just talking about, because it's really the kind of safety window that we're concerned about rather than the micromolar value. So I'll just do a brief overview of how we kind of generated the human dose predictions, and then I can kind of go into the HERG safety window. So we used PBPK software. So we have Simcyp Discovery PBPK software, which is kind of the gold standard approach really to generating human dose predictions. So we kind of derived the clearance and volume of distribution values through a mixture really of kind of in vitro and vivo extrapolation, allometric scaling and in silico methods, and really wanted to get confidence in those values that we're using by getting similar values across different methodologies. So we generated a clearance value of about 5.5 mil per min per kilogram, which is around 25% liver blood flow, and a volume of distribution of about 4.5 litres per kilogram. So we kind of plugged this into the software and allowed it to kind of predict the other kind of parameters and the PK profile. So we did this for adults. So the software uses a body weight of about 81 kilograms for an adult. And then we scaled that down to children using a body weight scaling method to then adjust for the predicted effective dose in children. So we use 5 and 10 years. So we've got about 200 mgs once a day in a 5-year-old and 360 mgs once a day in a 10-year-old. What we're really interested in when we're looking at the safety window is what is that anticipated effective free Cmax? So what's that maximum free concentration that we're going to derive from these dosing these patients, and how does that compare to the HERG safety window? So for these calculations, we used the most conservative estimate of HERG to give kind of almost a worst case scenario. So we use that 6.4 micromolar value. As Peter said, it does range anywhere up to about 18 micromolar. I think the key here is that it does have kind of a signal in the HERG assay, and how we can compare that to the anticipated free Cmax. So we took the anticipated effective total Cmax, which was generated in the PBPK software, which was 3.9 micromolar, and adjusted that for the human plasma protein binding, which was generated in a rapid equilibrium dialysis in vitro assay, which gave an unbound fraction or FU of 0.059. So essentially multiplying those values together, which gave us an anticipated effective free Cmax of 0.23 micromolar, which you can kind of see on this graph on the left-hand side. So that blue line there is the free concentration in nanomolar of the plasma, and you can see that kind of Cmax sits around that kind of 200 nanomolar level. So that's what we're kind of looking at in terms of free Cmax. So when we compare that to the 6.4 micromolar HERG value, we get a safety window of about 28-fold. Obviously, this is our most conservative estimate, so this is kind of our worst case scenario. And as Peter said, we kind of look to a 30-fold safety window, or a lot of companies look towards that 30-fold safety window. So depending on the indication and the disease, it can go up to maybe 100-fold, for example. But actually 30-fold is generally considered a suitable safety window, especially in these kind of indications. So we're kind of there or thereabouts that 30, and it kind of improves as you look at the different HERG values. So I think what we can kind of summarize is that, yes, we do have what is potentially on paper a poor HERG IC50 in the kind of single micromolar value. However, we need to put that into context of what we're expecting to see when we go into the clinic. And that safety window we would consider as okay and not a massive red flag. However, we may want to start looking at thinking about in vivo telemetry studies to look at that kind of cardiac risk as we go in vivo, and kind of pull those two data sets together to get a better idea of the risk. So I appreciate that's a bit of a whistle-stop tour, but if anyone has any questions or comments, then please go ahead.
Liz, hi, it's Mike. You've got access to the PBPK modelling, and I'm aware that there's been some emerging data around enzyme phenotyping which suggests there's appears to be a low involvement of cytochrome P450s in the metabolism of this compound. So that sort of may suggest there may be lower variability obviously driven by environmental or genetic differences in the expression of metabolic enzymes in various patients. But within your PBPK software, is there anything you can do at this time to sort of look at the potential PK in a population of subjects, or do you need more of that enzyme phenotyping data to help you pick those populations?
I would need to go back and check, so we don't have the full bells and whistles package.
Okay.
However, I think we can put in the phenotyping. I think what we would want to do is get some kind of estimate of renal or biliary clearance that we can put in there as well, so that we get kind of fuller picture of the excretion of the compound. But we can put in there some of the phenotyping data and see how that influences things. As I say, the biliary and renal, if we could get some kind of preclinical value there, that would help in terms of building the model. But yes, I think I would need to go and double check, but I think it's something that we can now incorporate into the model just to see how it affects things.
So do you have a rat bile duct cannulation study in the plans somewhere? Is that something that's been lined up? Then you'll get that renal elimination you can put into context of, you know, clearance, creatinine clearance rates, plasma protein binding, understand if it's just passive or perhaps actively driven, at least in a rodent species, and get some evidence of biliary elimination of parent compound. Is that something that we've got?
I think that is
to help you. Yeah. Okay.
From what I've seen.
Mm.
But as you say, it may be that there's some kind of active mechanism going on there, because obviously renal tends to be relatively low based on free fraction. So it may be that there's an active component there as well to the excretion that we can look into, especially as we know it is a substrate for PGP as well. So it may be that there are kind of other mechanisms kind of working to pump out the compound.
Liz, do you have any comment, or maybe anyone else on the line has a comment on how we present this kind of moving forward? Do we go with the worst number that we have, which is this one from Pharmaron, or is a mean of the four runs that we have something that we should consider?
I think what we're doing internally at the moment is just trying to understand the differences between those assays, to try and understand why that level of variability is there. I think 3 of the values are quite within a very small range, and then there's kind of the 18 micromolar one, which is a bit higher. I think we're just trying to understand, is it the same batch, same level of purity, for example, that have gone into those assays? So we're just kind of exploring that internally first. Based on the ICH guidelines, it uses kind of confidence intervals, and looking at a 95% confidence interval, which takes into account the variability across the assays. But the precedence tends to be kind of the most conservative estimate. But I think it's something we could seek advice on. Especially within the CRL, we have kind of the safety advisory service, scientific advisory service that we can kind of lean on and maybe get their advice, and some of our colleagues internally to see what their kind of view on this is, because as you say, I think there are different ways that we can approach it. I don't know if anyone on the line has any kind of thoughts about that, because that would be interesting to hear as well.
And I think recently people are more interested in cardiomyocyte data. I mean, you know, HERG had its precedence because it's just one parameter, is IKR, but people are now starting to look at more parameters to sort of de-risk development, because everybody is aware, like, you know, you can have a HERG channel blocker, but if you're also blocking calcium channel, it's kind of just, you know, mitigate that effect. So, and I don't know if you've done all of the channel works on 209 to see if there's any other channel activity that might mitigate that HERG liability, and so you may not have to worry about that too much, but looks like people are now drifting more towards cardiomyocyte data. And I think we did generate some of that for 209, didn't we?
We did Kevin now.
Yeah. I was looking when I saw some of the internal emails internally at Charles River. I went and had a look in the sort of Dotmatics sort of browser page, and there is data there from a SIPA panel, which would have been run at Charles River. So that is some of those other ion channels, but what the numbers were, and I'm not expert enough to be able, I said, oh, it's been done. And I think one of those Charles River HERGs was as part of that broader panel. But I think that the purity matter that sort of Liz talked about is an interesting one. Let's just try and understand what those batches were that went into those assays, what we knew about purity. Because obviously, as compounds tend to sort of progress into the later stages and towards preclinical development, you're tending to get better quality, purer material because you're lining up for the safety studies. And there may be an argument to say when some of that becomes available to put it back in, if we can understand what was happening historically, you know, I used to say to projects, wait until you do your CYP-TDI assay till you've got your best batch of compound, because otherwise it just becomes a bit of a purity check. So you want good quality material for these assays.
I do know that the Pharmaron batch is greater than 99%,
Okay,
and I believe that's the same batch that was run for the last one at Charles River, which was 18. Now, the two earlier ones back from 2018 and 2019, I'd have to go back. I'm not sure which batch those were run with and what the purity of those two were, but yeah, it's kind of, we've got the lowest and the highest, I believe, with the same batch of material.
The same batch. So the protocols, I mean, if we can get the protocols, Liz.
Yeah, because the 80 micromolar
temperature
came out of the broader SIPR panel. So it wasn't in kind of the isolated HERG one where we looked at CAB and NAB as well in that SIPR panel, and that's where the 18 micromolar came from. So that may be a slightly different study design potentially. So we would need to kind of look into that.
And check the temperature of the assays, because I'm aware sometimes some of the higher throughput assays are not done at physiological temperature. I think they've caught up now in terms of this automation, things like that. Obviously, the manual patch clamp assays, they had more control over assay temperature. So it would be worth getting the protocols like those two assays where we got the same batch, which is very good purity, with different numbers, and just see if there's anything in the terms of assay temperature, for example, or that may help understand.
And I mean, on one other thing too, like if you really think we have a HERG issue, I mean, usually the next step up is to do telemetry, dog telemetry and stuff. I mean, so I don't know if that's part of the plan for your development strategy to, you know, just do a more advanced assay to kind of just mitigate that risk.
Yeah, I think we'll consider that. We asked that with the Tessa Jowell, and I think you know, we can assess that as part of the tox study.
Okay.
Okay.
So just a question. Sorry, before we get there then. So do we have cross-species plasma protein binding? And what I'm thinking is, in my experience, one of the models where you can do in vivo, you know, cardiovascular de-risking is they do what they call the ramped infusion study in dogs. So you go in at a certain concentration, you do a steady state infusion for a while, monitor, push the infusion rate up, leave it reach steady state again, and you just sort of do this stepwise increase. But I'm just thinking, as we've got dog PK on the plans, it might be worth making sure we've got plasma protein binding in the dog so we understand what the likely free concentrations are, so we can then start to understand quite quickly, you know, where free exposures may be from an intravenous dose perspective in downstream studies, for example. You know, just sort of starting to think downstream, because it'll all be driven by free concentration. This will be part of a safety pharmacology package when you start to do these types of studies. But once you start to go in vivo, one of them is this sort of ramped infusion in a dog is one of the models I'm aware of.
Yeah, we have plasma protein binding in three species, but I can't recall if it's mice, rat, dog.
We've got mouse, rat, and human. So
mouse, rat, and human. Okay.
Dog.
So yeah, I couldn't find any that I think. So maybe that's something to backfill. Just have that available, especially if the plans are progressing in terms of design of the sort of dog PK study. So we can all read out at the same time. And I would say, given the good pharmacokinetics of this compound in the rat, we probably don't need to go in at a dose of 10 mg per kg orally in the dog. You can probably go lower at something like 3 mg per kg, because if it's got a low to moderate clearance, you're going to have enough drug on board at 3 mg per kg to get a measure of oral bioavailability. So I think evidence to date has been it's got pretty good oral pharmacokinetics. So I think 3 would be a good enough dose, but that's something obviously the study directors can recommend when they see some of the metabolic stability data. Zoom Pharma are doing the work.
I think it's 3 that we're going.
Yeah.
Okay.
And this is really just a summary. So we kind of met with some of our scientific advisory services teams who have kind of dealt with a lot of these kind of compounds going into development to look at the kind of toxicology and clinical planning. I think it probably aligns with a lot of what we've heard from Tessa, Jowell, and it was really about how do we approach the toxicology plan? Do we use juvenile animals for toxicology, or do we need to use adult animals? And it was really, if there's no intention to go into adults in the clinic, then it's not that helpful to run toxicology in adult animals. So it'd be recommended to use the juvenile animals for the toxicology studies, which would need to be used pretty much immediately after weaning. But obviously this very much depends on the clinical plan and what the agreement is there, and then we can kind of work backwards. And they also mentioned that normally in these kind of studies we'd look at the kind of no observed adverse effect level, and they said actually in this you're really looking for the highest non-severely toxic dose in the dose escalation studies, which is kind of looking at lethal dose basically, because of the nature of the disease and of the drug. They also recommended collecting the brain samples in the toxicology studies to get an idea of the brain kinetics and any kind of accumulation, and also to consider any potential for acquired resistance through efflux mechanisms. I think we've kind of looked into this a little bit as well since, and we don't think that this is necessarily going to be an issue based on what's in the literature and just the length of the disease, but just something to consider. And also something we touched upon before is just thinking about the biomarkers that will be used in the clinic. So this is really just a summary of their kind of thoughts on the kind of toxicology and clinical planning. But as I said, I think it aligns quite well with kind of other advice that's been given from kind of external sources as well. But yeah, if there's any questions or comments, please go ahead.
Okay, we are coming up to the top of the hour. Just kind of go through this quickly. We've got a couple of studies in progress right now. So this data will be coming out in the next few weeks. And as I mentioned at the beginning, we're basically in the contract and review phase for the salt screen and the scale up. So I just want to know if we want to switch this around. Jerome, I know you know we want to have your input, whether or not you have time to stick around for a few minutes past 10, or do you want to maybe discuss your model now? If anyone needs to drop off, fully understand, but continue on to finish this off for whoever's remaining to be available. But maybe I'll just turn it over to Jerome right now.
Yeah, thanks. I'll try to be quick because I have another meeting coming up anyways. But yeah. So essentially, my lab has a grant from the DIPG collaborative, the Cure Starts Now, to really look at ACVR1 mutant diffuse midline glioma, and we're particularly interested in knowing what the ACVR1 mutation does mechanistically, and then find ways to counteract its activity. So I have a few slides, but maybe that given the timing, it's I'll just kind of summarize what our approach is. So we have a mouse model which combines histone 3K27M, ACVR1 G328V. ACVR1 is ALK2, just for everybody, and then PIK3CA. mutation. So that's a combination that's very often seen in patients. And these mice develop very aggressive brain tumors. They survive until maybe 60 days after birth. So it's a really quick model. And each of the three mutations, histone, ACVR1, ALK2, or NP3C, contributes to the phenotype. So if we only have two of them, sometimes we get tumors, but it takes longer. Okay, so we think it's a good model to really look at what the ACVR1 mutation does in a kind of like physiologically relevant context. So what we're about to do is compare tumors that have ACVR1 mutations versus those that don't using single-cell transcriptomics. So that's one thing that we're doing, but the obvious complement to that would be to look at our ACVR1 mutant tumors with and without inhibitors, right? And so we thought that probably the best thing to test would be the lead M4K compounds, which I understand is M4K 2009 at this point. And so we would take a few mice and administer them with the compound. So there's a lot of things that we can do. We could do a survival study, but molecularly, we want to complement our single-cell transcriptomic studies with similar analyses in drug-treated mice. So we would take a few animals treated with drug and then profile them at single-cell with single-cell transcriptomics, profile their tumors. I mean, or their brain if the inhibitor wipes out the tumor, which probably won't happen. So the question is, how do we go about to do this in vivo administration, right? So how much drug do we give for how long? What's the vehicle or the solvent, and can we have drug? Basically, that's essentially what it is.
So yeah, I think we can certainly discuss this further, but how much material would you need? So we do have probably 3 to 4 grams of the HCl salt right now. I think that we could use in this study. So that's not a problem. Maybe you don't even need that much.
And then how long should we treat the animals? What kind of dosing and how long do we treat the animals for?
So we've been dosing in 0.5 methylcellulose at 50 mg per kg. Okay. In the Jones model, we've been dosing them for weeks. At that 5 days on, 2 days off.
Yeah. I mean, of course, in our model, we survival is it's about in the controls, right? Like what we would expect for vehicle treated would be something between 50 to 80 days after birth. So that's after birth. I think we would not necessarily start the treatment from birth, right? We would probably wait maybe 4 weeks after birth. This is gavage that the drug is given.
Yeah.
Yes.
Yeah. Sometimes it's hard to gavage mice before, I would say, 5 weeks after birth. At least in our other studies, it's just more difficult and to do it with the mice. So maybe we have to consider using smaller gavage needles. But yeah, so essentially the duration of the treatment realistically would be maybe 4 weeks. Maybe we could go for, or something like that, 3 to 4 weeks or something like that. The control mice get, pretty bad pretty quickly. So there's always the question also about, well, can they tolerate the drug administration?
Right.
And how many?
For the project, we're actually talking with a company in the US that can actually put the drug in food, but something that we could potentially consider too. Yeah.
How many mice you're looking at, Jerome, in each group?
So it depends what we want to do. For survival study, it would be more. For single-cell transcriptomics, we would do probably we would start with 4 animals. So we would treat more than 4 in order to be able to get sufficient material, because we would use basically this technology from 10x Genomics, which enables on-chip multiplexing, so we can actually profile 4 tumors simultaneously in a relatively cost-efficient manner. And so, yeah, so 4 controls and 4 treated for the transcriptomics. So if we wanted to do a survival study or additional, molecular profiling, we could do more animals for sure. These animals are generated by breeding, right? It's all endogenous mutations, spontaneous tumors. It's a genetically engineered model. So there is also that component of it. It's if you think of the breeding, we need 4 alleles, right? So the 3 mutations, they're all heterozygous, and the Cree driver, because it's conditionally targeted to a specific cell population. So we, from a typical breeding, we have 1 out of 16 animals that has the target genotype. So there's also the question of generating the sufficient number of animals for the studies in this in more or less simultaneous cohort. That can be a challenge.
Well, I think as for the compound, I think as what Pete just said, we should have enough compound. I was just doing the math. Even if we do for 10 animals, 5 days, and for 4 weeks, we should be able to do it like under 1 gram compound. So should be enough compound. We should have enough compound.
Easily, yeah.
Yeah.
So yeah, so we can talk about how to obtain that and set that up. Yeah, what we're doing right now, we're going to do soon that first leg of the study, which is to compare ACVR1 mutant versus ACVR1 wild type tumors. And that's going to also provide us, hopefully, a good baseline to interpret any effect that we see of the inhibitor, because we would have like a scenario where we have no hyperactive ACVR1 signaling versus the gain of function mutation. So that could help interpret whatever we see in the drug studies. The one thing I saw in today's meeting that I just kind of wanted to go back to was like I guess the general consensus is that this compound really gets well into the brain, or what's the take home there?
It goes well. It goes more than 30% of the plasma concentration goes in the brain approximately. So it's not bad.
Okay, all right. I just wanted to clarify that, because of course that's a big, it's a big thing in mice, right? Because we're going to deal with mice, right? So, if it's a compound that we're not even sure gets into the brain, then I'm like, well, maybe it's not a good idea for an experiment. But
yeah, no, we certainly have that data. I'll send you that. You can take a look at the blood brain to plasma ratio, as well as the KPUU that we've calculated for the efficiency which this compound gets into the brain.
And just to give an idea, if we were to do a survival study, what we've seen is that if we compare the mice that have the ACVR1 mutation versus those that don't, the ones that have the ACVR1 mutation, I think the median endpoint survival, the median survival duration is about 60 to 70 days after birth. Without the ACVR1 mutation, it's like 120 to 130 days. So that would be the maximum sort of, like, if we have a compound that completely blocks mutant ACVR1 signaling and works like, perfectly well, that's the kind of survival benefit we would expect in that model.
No, that'll be interesting to see for sure. Yeah, so we can arrange to ship out compound, whenever you're ready to start a study.
Yeah, absolutely. We're generating those mice all the time, so we should be able to plan that soon. Yeah.
Yeah, so I'll reconnect with you later this week, and we'll arrange to have some material shipped out.
Great.
Thanks, Jerome.
Thank you.
Ahmed, maybe you want to give an update on some plans we have for imaging using mass spec.
Yeah, great. We are very quickly. I know we are running out of time here, or we are already out of time. So basically, what we are, I met a team from Spain. They are doing mass spec imaging and they can get the imaging resolution almost down to 10 micron. And what they are going to do after dosing the compound, they are going to look at the compound concentration in different parts of brain, so we'll get this spatial distribution of the compound. So that's what is going on. They are optimizing the method for 209 and 2281. So the optimization is done. Now we are going through the animal availability and how they can dose the animals and get the brain from there. So that's ongoing. So hopefully we'll get some data early January. So we'll discuss when we get the data.
Very cool. Yeah, so just to any questions on that study? So just to end off, I wanted to mention this meeting we had with the Connect Consortium where we kind of discussed what a clinical trial might look like. And, you know, at least at the beginning, they emphasized that we really should continue to look at combination studies, which we certainly will. But the challenge is what is a trial going to look like? And so some of the scenarios that we discussed would be one kind of a typical trial where you went into healthy adult patients initially. And obviously I would say this is rare for an oncology agent. And the upside is that's the traditional route. There'd be, much less regulatory pushback maybe using that route. But the downside is we wouldn't have support from Connect for that type of trial. So we would internally have to raise significant funds and basically, run that trial ourselves. So that would be a significant challenge for, the situation we're in. So we have to kind of weigh that as, if we have to go that route, it's probably not going to happen, quite honestly. The second scenario was a phase 1/2 in young children. And the advantages here is, that age group has the highest potential to benefit from early intervention with an ALK2 inhibitor. Now, the big challenge there would be ethical concerns because you'd have to kind of go in at a subtherapeutic dose to begin with and then dose escalate. So obviously that is, something that we have to be, aware of and the ethical implications of something like that. Now, the third scenario, which I think is probably the most likely, provided we can have justification of broadening the eligibility for that initial trial to include groups of patients that might not have had a biopsy, so new patients or newly diagnosed patients, as well as recurrent patients that have kind of gone through a standard of care already. And that cohort would be done in older patients, so 12 and up to young adults. And with that scenario, then you could, once you get a dose, you've identified a dose level and schedule, you could potentially have a age de-escalation down into a phase 2 to include those younger patients. So I think these are some scenarios that we discussed internally, but we really kind of need to start planning for that and then put this in front of a regulatory agency to get their guidance on what the best path forward is. So I think my plan right now is to go back to Connect Consortium and see if they're able to facilitate that type of meeting on our behalf that we could certainly attend. And if they can't, then we'll have to look at finding a consultant or regulatory group that can kind of help facilitate setting up that meeting and putting together that, at least at an initial clinical plan that we can have the back and forth discussion with the regulatory agency to see what is going to be the best path forward. I think in at least with the Connect Consortium, there's a lot of support for getting this into children as quickly as possible. So first in child, there's still a lot of support for that. So I think that's what we kind of have to go and talk to the regulators about. So that's all I have from our side. Any other questions or comments before I let everyone get on with their day and get on with their holidays, because we'll probably meet again in January, unless there's an urgent need to meet beforehand.
Sounds good.
Okay.
Thank you, Peter.
Thanks a lot.
everyone.
Thank
Bye-bye.
you.
All right.
Bye.
We'll talk to you soon. Bye.